Hi, today we're going to be talking about DNA, the genetic material of life. DNA is composed of two chains of nucleotides that are coiled together to form a double helix. There are 4 nucleotide bases in DNA. Nucleotide bases of opposite strands form hydrogen bonds with each other in a process called complementary base pairing. In complementary base pairing in DNA, the bases adenine and thymine are bonded together. Guanine and cytosine are also bonded together. The structure of a nucleotide is quite simple. There's a phosphate group, a pentose sugar, and a nitrogenous base. Now let's segue into DNA replication.
DNA replication. Oh yeah.
In DNA replication, DNA helicase comes and unwinds the DNA into 2 separate strands. Single-stranded binding proteins then attach themselves in order to keep them apart. Then, DNA primase comes and puts an RNA primer in place.
RNA primer.
DNA polymerase 3 comes and begins DNA synthesis, forming complementary pairs with the original strand. There are 2 strands from the original DNA molecule, the leading strand and the lagging strand. Since DNA can only be made in a 5-3 direction, DNA polymerase must synthesize DNA in short Okazaki fragments.
Okazaki.
DNA polymerase 1 then comes and turns the RNA primer into DNA. Then, DNA ligase comes and connects the Okazaki fragments, completing the DNA molecule. At this point I got too lazy, so I decided not to use the whiteboards and I decided to just record it on my computer. So that's why it looks different. Okay. Yeah. Now let's talk about DNA transcription, where RNA is produced from a DNA template. The first step of transcription is initiation. In initiation, transcription begins with RNA polymerase binding with a region of DNA called a promoter. The promoter is responsible for defining the start and direction of transcription, as well as the strand being transcribed. From this point on, the second step of transcription, elongation, occurs.
Oh yeah.
In elongation, RNA polymerase reads down the DNA molecule until a stop sequence is eventually reached. Once the stop sequence is reached, the third step of transcription, termination, occurs. In termination, RNA polymerase releases the mRNA molecule and closes the DNA. Unfortunately, this mRNA molecule is actually RNA transcript or pre-RNA and must be filtered. Pre-RNA is comprised of exons, which code for proteins, and introns, which don't. In order to remove the introns, a spliceosome, which contains small nuclear RNA, comes and identifies the introns through complementary base pairing, and then kicks them out. The spliceosomes are then able to leave and go home to their families. The RNA molecule then comes together to form a proper mRNA.
mRNA, baby.
Now the mRNA is ready to leave the nucleus and go out into the real world. I mean, the cytoplasm. Here, the mRNA finds a ribosome. This is when translation begins. The first step of translation is initiation. In initiation, the mRNA binds to the small ribosomal subunit of the ribosome. This occurs when a start codon is read. The ribosome has 3 binding sites: the exit site, the peptide site, and the amino acid site. Then, an initiator tRNA molecule binds to the peptide site, carrying an amino acid. Now it is time for the second step, elongation. This is where the large ribosomal subunit comes into play. Another tRNA molecule carrying another amino acid binds to the A site. The 2 amino acids are then linked with a peptide bond. The ribosome then moves forward and the tRNA in the exit site leaves.
Bye bye.
Now, a new tRNA molecule enters the amino acid site and a peptide bond is formed between the amino acids. This process continues, and the polypeptide continues to grow larger until it eventually looks like this. Polypeptide synthesis eventually stops, and a release factor is placed instead of tRNA. This occurs at a segment of mRNA called a stop codon. This is where the final stage of translation occurs, called termination. In termination, the polypeptide leaves the ribosome and is finally free to fold into a protein.